Recording stopped. Recording in progress.
Universitas Gadjah Mada, bring ideas to life and make a difference. As an inclusive university, we believe in a world-class education system for all. From the struggle and courage of the Indonesian Revolution for independence, we started a journey to build the nation. We have since laid the foundation for lifelong learning that extends across generations. As we face rapid cultural and technological shifts, we understand that staying one step ahead of challenges is the way forward. Innovation and transformation are integral to every move we make, including how we deliver education. In response to industry challenges, we are always open to feedback and collaboration. We translate research on food security, sustainable energy, net zero healthcare into useful products and formulate solutions to societal needs and problems, as seen from our role and contribution to tackling the COVID-19 pandemic. Our new, ever-developing infrastructure with a variety of supporting systems and modern technology is built to accelerate the pursuit of answers while making everyone feel at home. Through our focus on food, health, and energy, we join hands to make the world a better place now and into the future. Here at UGM, passions are nurtured beyond academic fields. The facilities that cater to their needs, students are given the space to unlock their potential, unleash their creativity, and reach for the stars. Our dedication to community development reaches far and wide to every corner of the archipelago and those closest to us. We synergize and share roles with various stakeholders to build an empowered and resilient community. Our commitment to serving the interests of the nation and humanity has led us to pick up achievements and awards at the individual, group, and institutional levels on the national and international stages. Unity in diversity. We push knowledge forward and make a high community impact that matters in harmony with our long-held principle, globally respected.
We are UGM.
Wana Gama dibangun dengan spirit yang menjulang tinggi pada tahun 1960-an. Pada saat itu, dengan kondisi sumber daya manusia dan teknologi yang terbatas, serta finansial yang lemah, mampu memulihkan hutan dan lahan kritis di Gunung Kidul. Universitas Gadjah Mada melalui Fakultas Kehutanan mengabdikan diri melalui Tridharma Perguruan Tinggi. Terbangunlah Hutan Pendidikan Wana Gama.
Hutan Pendidikan Wana Gama adalah hutan pendidikan yang dimiliki oleh Fakultas Kehutanan UGM seluas 625,25 hektar yang merupakan hutan pendidikan yang dibangun mulai dari lahan kritis, sekarang telah menjadi hutan yang hijau dengan segala macam fungsinya, baik untuk penyedia oksigen, air, tempat rekreasi, dan juga tempat pendidikan.
Wana memiliki makna hutan dan Gama memiliki kepanjangan dari Gajah Mada. Wana Gama merupakan karya Universitas Gadjah Mada melalui mengukir alam dengan rehabilitasi lahan kritis.
Wana Gama itu dibangun dengan spirit yang sangat jelas, rehabilitasi lahan kritis untuk kesejahteraan masyarakat dan rehabilitasi lahan kritis untuk penyelamatan lingkungan. Model ini sudah selayaknya dijadikan sebagai contoh untuk rehabilitasi lahan kritis di Indonesia yang saat ini luas lahan kritis lebih dari 40 juta.
Wana Gama telah menjadi rujukan rehabilitasi hutan dan lahan kritis, dan saat ini spektrumnya terus meluas tidak hanya di Gunung Kidul, kawasan Geopark Gunung Sewu, namun juga Indonesia dan bahkan dunia. Hutan Pendidikan Wana Gama saat ini dikelola secara profesional oleh Sivitas Akademika Fakultas Kehutanan Universitas Gadjah Mada melalui unit pengelolaan Wana Gama.
Pengelolaan Wana Gama terus menjunjung tinggi spirit perjuangan Wana Gama, sehingga Wana Gama menjadi sumber inspirasi dalam proses berpikir dan sebagai tempat belajar mengolah ekosistem alam. Salam Wana Gama, salam harmoni kehidupan dengan alam.
Kegiatan pendukung Tridharma Perguruan Tinggi di Wana Gama antara lain kebun pembibitan, percontohan budidaya ulat sutra, dan museum kayu. Kegiatan di Wana Gama sebagai rujukan rehabilitasi hutan dan lahan kritis antara lain pendidikan, pelatihan, penelitian, dan pengabdian masyarakat. Wana Gama sebagai hutan pendidikan digunakan sebagai kampus lapangan dalam mendidik mahasiswa Fakultas Kehutanan Universitas Gadjah Mada. Pelatihan-pelatihan yang dilaksanakan di Wana Gama diikuti oleh peserta dari dalam maupun luar negeri. Jati Mega merupakan salah satu buah karya penelitian yang dilaksanakan di Wana Gama. Wana Gama juga memfasilitasi masyarakat umum yang ingin menggunakan kawasan Wana Gama untuk kegiatan wisata alam. Penginapan yang sangat memadai sudah tersedia di Wana Gama. Masyarakat dapat menikmati kenyamanan fasilitas penginapan di Wana Gama dengan harga yang sangat terjangkau.
Yayasan UMI itu maksudnya itu untuk membackup pengelolaan Wana Gama. Dia memesankan kepada kita semua untuk supaya tidak meninggalkan Wana Gama, maka dibuatlah Yayasan UMI yang pertama kali modalnya kita peroleh dari Bu Umi sendiri dan dari saudara-saudaranya dan dari beberapa orang yang lain. Kemudian didirikanlah Yayasan UMI. Selama ini kami hanya membantu bagaimana melancarkan pekerjaan-pekerjaan teknis di Wanagama. Kami juga bisa mengatur supaya dana itu tidak habis ya, biar bisa berkembang. Jadi kami tetap akan berada di Wanagama untuk sesuai dengan pesan dari almarhumah yang menjadi perintis dari Wanagama.
Bagi masyarakat yang memiliki hobi bersepeda, dapat mencoba wisata bersepeda mengelilingi hutan Wanagama. Untuk memperoleh tantangan yang lebih ekstrim, pesepeda dapat mencoba trek khusus bersepeda di Wanagama. Dalam upaya menumbuh kembangkan spirit Wanagama kepada generasi Indonesia, maka Wanagama diarahkan sebagai Eco-Edu Forest. Salah satu upaya awal untuk menginisiasi Wanagama sebagai Eco-Edu Forest yaitu dengan melakukan pengembangan program Wanagama 1000 Selfie atau WG1000S. WG1000S sebagai wahana untuk menyebarluaskan spirit Wanagama lintas generasi sebagai bagian penting dari pendidikan lingkungan dan upaya berkelanjutan untuk menjaga kelestarian ekosistem sumber daya alam.
Jadi pijakan pasti, ilmu dan aksi wujudkan janji, kolaborasi, soliditas membara untuk hutan lestari selamanya. Bersama alumni bersatu langkah menatap masa depan yang cerah. Diesna Alis ke-62, Fakultas Kehutanan UGM tercinta, reinventing pendidikan kita, menjawab tantangan Indonesia. Hijaukan negeri, semangat membara, demi bumi kita bersama. Langkah maju, hati menyatu, jaga hutan, jaga bumi biru, cerah harapan, masa depan hijau, bersama kita takkan mundur jauh. Diesna Alis 62, Fakultas Kehutanan UGM tercinta, reinventing pendidikan kita, menjawab tantangan Indonesia. Hijaukan negeri, semangat membara, UGM jaya selamanya. Menjaga bumi ini dari kampus hijau di Bulak Sumbu untuk hutan lestari yang makmur. Bersama alumni bersatu langkah menatap masa depan yang cerah. Diesna Alis 62, Fakultas Kehutanan UGM tercinta, reinventing pendidikan kita, menjawab tantangan Indonesia. Hijaukan negeri, semangat membara, demi bumi kita bersama. Dari kampus hijau di Bulak Sumbu untuk hutan lestari yang makmur. Bersama alumni bersatu langkah menatap masa depan yang cerah. Diesna Alis 62, Fakultas Kehutanan UGM tercinta, reinventing pendidikan kita, menjawab tantangan Indonesia. Hijaukan negeri, semangat membara, demi bumi kita bersama. UGM lestari selamanya.
Universitas Gadjah Mada bring ideas to life and make a difference. As an inclusive university, we believe in a world-class education system for all. From the struggle and courage of the Indonesian Revolution for Independence, we started a journey to build the nation. We have since laid the foundation for lifelong learning that extends across generations. As we face rapid cultural and technological shifts, we understand that staying one step ahead of challenges is the way forward. Innovation and transformation are integral to every move we make, including how we deliver education. In response to industry challenges, we are always open to feedback and collaboration. We translate research on food security, sustainable energy, net zero healthcare into useful products and formulate solutions to societal needs and problems, as seen from our role and contribution to tackling the COVID-19 pandemic. Our new, ever-developing infrastructure with a variety of supporting systems and modern technology is built to accelerate the pursuit of answers while making everyone feel at home. Through our focus on food, health, and energy, we join hands to make the world a better place now and into the future. Here at UGM, passions are nurtured beyond academic fields. The facilities that cater to their needs, students are given the space to unlock their potential, unleash their creativity, and reach for the stars. Our dedication to community development reaches far and wide to every corner of the archipelago and those closest to us. We synergize and share roles with various stakeholders to build an empowered and resilient community. Our commitment to serving the interests of the nation and humanity has led us to pick up achievements and awards at the individual, group, and institutional levels on the national and international stages. Unity in diversity. We push knowledge forward and make a high community impact that matters in harmony with our long-held principle, globally respected.
We are UGM.
Wanagama dibangun dengan spirit yang menjulang tinggi pada tahun 1960-an. Pada saat itu, dengan kondisi sumber daya manusia dan teknologi yang terbatas, serta finansial yang lemah, mampu memulihkan hutan dan lahan kritis di Gunung Kidul. Universitas Gadjah Mada melalui Fakultas Kehutanan mengabdikan diri melalui Tridharma Perguruan Tinggi. Terbangunlah Hutan Pendidikan Wanagama.
Hutan Pendidikan Wanagama adalah hutan pendidikan yang dimiliki oleh Fakultas Kehutanan UGM seluas 625,25 hektar yang merupakan hutan pendidikan yang dibangun mulai dari lahan kritis sekarang telah menjadi hutan yang hijau dengan segala macam fungsinya, baik untuk penyedia oksigen, air, tempat rekreasi, dan juga tempat pendidikan.
Wana memiliki makna hutan dan Gama memiliki kepanjangan dari Gajah Mada. Wanagama merupakan karya Universitas Gajah Mada melalui mengukir alam dengan rehabilitasi lahan kritis.
Wanagama itu dibangun dengan spirit yang sangat jelas, rehabilitasi lahan kritis untuk kesejahteraan masyarakat dan rehabilitasi lahan kritis untuk penyelamatan lingkungan. Model ini sudah selayaknya dijadikan sebagai contoh untuk rehabilitasi lahan kritis di Indonesia yang saat ini luas lahan kritis lebih dari 40 juta hektar.
Wanagama telah menjadi rujukan rehabilitasi hutan dan lahan kritis, dan saat ini spektrumnya terus meluas tidak hanya di Gunung Kidul, kawasan Geopark Gunung Sewu, namun juga Indonesia dan bahkan dunia. Hutan Pendidikan Wanagama saat ini dikelola secara profesional oleh Sivitas Akademika Fakultas Kehutanan Universitas Gadjah Mada melalui unit pengelolaan Wanagama.
Pengelolaan Wanagama terus menjunjung tinggi spirit perjuangan Wanagama, sehingga Wanagama menjadi sumber inspirasi dalam proses berpikir dan sebagai tempat belajar mengolah ekosistem alam. Salam Wanagama, salam harmoni kehidupan dengan alam.
Kegiatan pendukung Tridharma Perguruan Tinggi di Wanagama antara lain kebun pembibitan, percontohan budidaya ulat sutra, dan museum kayu. Kegiatan di Wanagama sebagai rujukan rehabilitasi hutan dan lahan kritis antara lain pendidikan, pelatihan, penelitian, dan pengabdian masyarakat. Wanagama sebagai hutan pendidikan digunakan sebagai kampus lapangan dalam mendidik mahasiswa Fakultas Kehutanan Universitas Gadjah Mada. Pelatihan-pelatihan yang dilaksanakan di Wanagama diikuti oleh peserta dari dalam maupun luar negeri. Jati Mega merupakan salah satu buah karya penelitian yang dilaksanakan di Wanagama. Wanagama juga memfasilitasi masyarakat umum yang ingin menggunakan kawasan Wanagama untuk kegiatan wisata alam. Penginapan yang sangat memadai sudah tersedia di Wanagama. Masyarakat dapat menikmati kenyamanan fasilitas penginapan di Wanagama dengan harga yang sangat terjangkau.
Yayasan Umi itu maksudnya itu untuk membackup pengelolaan Wanagama. Dia memesankan kepada kita semua untuk supaya tidak meninggalkan Wanagama, maka dibuatlah Yayasan Umi yang pertama kali modalnya kita peroleh dari Bu Umi sendiri dan dari kemudian didirikanlah Yayasan Umi. Selama ini kami hanya membantu bagaimana melancarkan pekerjaan-pekerjaan teknis di Wanagama. Kami juga bisa mengatur supaya dana itu tidak habis, bisa berkembang. Jadi kami tetap akan berada di Wanagama untuk ini sesuai dengan pesan dari almarhumah yang menjadi perintis dari Wanagama.
Bagi masyarakat yang
Hello, Professor Grill, Professor Yamamoto, how are you? Thank you very much.
Halo.
Hello.
Assalamualaikum.
Waalaikumsalam.
Waalaikumsalam.
Thank you for attending this no, event.
You're welcome.
And thank you also the Dean, Dr. Sigarto, our Dean of Faculty of Forestry.
Oh, hello,
Halo,
everybody.
halo. Good evening. Good afternoon. Sorry.
Okay, and the Master of Ceremony, Mrs. Nifa, please.
Still 6 minutes, Pak Widi.
Yeah, some announcement about the event.
Yeah. Good afternoon from
I come back in a
Jakarta,
few
ladies and
minutes.
gentlemen. Yeah, this second webinar of plant biomechanics will start at 2 p.m. UTC +7 or West Indonesian time. While waiting, please use the official virtual background that the organizing committee has shared in the chat box. Thank you for your patience. And this is a little bit of announcement from our organizing committee of about the previous webinar series. You can access this YouTube channel. So for the participant who hasn't been present in the previous webinar, you can access in this recording. And also, we also would like to invite you to participate in our upcoming session, which is the 11th International Plant Biomechanics Conference, which will be held in Yogyakarta, Indonesia, on August 23 to 26, 2006. Okay. Thank you for your attention.
Dr. Yu Chen Long, good afternoon.
Hello, good afternoon. How are you?
I'm fine, and thank you very much to join us here.
Thank you very much for the invitation. Yeah.
Yeah. Good afternoon from Yogyakarta, Indonesia, and welcome to the distinguished guests, esteemed speakers, and participants from around the world to the second webinar series of Plant Biomechanics 2025, Road to the 11th International Plant Biomechanics Conference 2026. This webinar series has been designed as a platform to share and discuss the latest research and application in plant biomechanics as we build the road towards the 11th International Plant Biomechanics Conference 2026, which will take place here in Yogyakarta on August 23 to 26, 2026. My name is Hani, and I'm honored to serve as your MC for today's session. And this session is organized by Faculty of Forestry, Universitas Gadjah Mada, Indonesia, as part of event series in 62nd Dies Natalis or Faculty Anniversary of Faculty of Forestry, Universitas Gadjah Mada. Before we start our session, I invite you to use the virtual background that we have prepared in this chat box throughout this event. And first of all, allow me to share our today's program. Firstly, is the welcoming remarks from the person of organizing committee, Professor Widianto Dwi Nugroho. And secondly, remarks from the Dean of Faculty of Forestry, Dr. Sigit Sunda. And thirdly, the main event of the webinar from our esteemed speakers, which will be guided by our moderator, Dr. Cipta. And finally, there will be closing and photo session. Without further ado, let's begin our session with welcoming remarks from the chairperson of organizing committee, Professor Widianto Dwi Nugroho. To Professor Widianto Dwi Nugroho, the floor is yours.
Okay, thank you very much, Mbak Hani. Ladies and gentlemen, distinguished speakers and participants, Assalamualaikum warahmatullahi wabarakatuh. On behalf of organizing committee, it is truly make it pleasure to welcome all of you to the second webinar of the Plant Biomechanics Series 2025 as the part of our journey towards the 11th International Plant Biomechanics Conference next year, 2026. Thank you very much for joining us today. This event was created as a platform to share knowledge, exchange ideas, and discuss new findings in our field. This is organized by the Department of Forest Product Technology, Faculty of Forestry, Universitas Gadjah Mada, Yogyakarta. And more than just a scientific forum, this webinar is also about building a community, connecting researchers across continents, strengthening collaboration, and encouraging young scientists to engage in this field. We are also delighted to see such enthusiasm and close participation from researchers, academics, students, and practitioners around the world. We are very proud that the first webinar has successfully more than 200 participants from more than 20 countries. And in this second webinar, participants came from 24 countries, representing various universities, research institutions, and also industries. Your presence is here truly our shared passion for plant biomechanics. And today, we are honored to host a distinguished lineup of speakers. At first, Professor Joseph Grill from Centre National de la Recherche Scientifique France. Thank you very much, Professor Grill. And the second, Dr. Yue Chen Long from the National University of Singapore. Thank you very much, Dr. Yue Chen Long. And also Mr. Rafi Pujasmara, the young researcher and our young staff in Faculty of Forestry, Universitas Gadjah Mada, Indonesia. They will share their valuable insight and experience that I believe will inspire all of us. I hope webinars will open up new idea inspire collaboration and strengthen our commitment to bridging plant biomechanics engineering for sustainable solution. And so on again, thank you very much for your joining us today. I wish you an inspiring and enjoyable webinar. And one important thing, we look forward to seeing you in the 11th International Plant Biomechanics Conference next year in Yogyakarta, Indonesia in August 2026. Thank you mas. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi.
Thank you very much, Professor Widi, for delivering the welcoming remarks. And next, we will move on to the remarks from the Dean of Bioforestry, Universitas Gadjah Mada, Dr. Sigit Sunarta. To Dr. Sigit Sunarta, the floor is yours.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good morning, good afternoon, distinguished speakers, colleagues, participants that joining us from around the world. On behalf of Faculty of Forestry, Universitas Gadjah Mada, it is my great pleasure to welcome all of you to the webinar series 2025, Road to the 11th International Plant Biomechanics Conference 2026. This webinar series marked an important step as we move towards hosting the 11th International Plant Biomechanics Conference here in Jakarta, Indonesia in 2026. We are honored to be part of this global community that has been growing steadily since the first conference was held in Montpellier, France in 1994. Plant biomechanics is an increasingly vital field that helps us understand how plants interact with their environment. These insights are not only fascinating from the scientific point of view, but also highly relevant to many practical areas, from sustainable forestry and agriculture to biomaterials, ecological restoration, and climate resilience. In the face of today's environmental challenges, including deforestation, land degradation, and the impact of climate change, understanding plant biomechanics give us tools to make better decisions for the future of our ecosystem. This webinar series was designed as an open forum to share ideas, discuss recent advances, and connect people working in this exciting field. Throughout the year, the series will bring together researchers, educators, and students, both experienced and emerging, to exchange perspective and strengthen collaboration. More than just a scientific program, this series is also community building effort. It's our hope that through this session, we can build momentum and inspire many of today's participants to also join us in Yogyakarta for the 2026 conference as speakers, contributor, or attendees. Allow me to say a few words about Yogyakarta, the city that will host the 2026 conference. Yogyakarta is not only home to the Universitas Gadjah Mada, one of the Indonesian leading university, but also a city known for its rich cultural heritage and deep connection to nature. Here, tradition and innovation live side by side. Visitors will find centuries-old temples like Borobudur and Prambanan, vibrant batik and silver craft workshop, and the warm hospitality that made Jakarta known as the city of culture and education. The landscape surrounding the city, from coastal forest to volcano mountain, reflect the beauty and diversity of Indonesia ecosystem. It is a place where the science culture truly meet, make it fitting venue for the upcoming planned Biomechanics conference. To all participants, I invite you to take part actively in today's webinar and in the upcoming session. Let us use this opportunity to share knowledge, strengthen international cooperation, and inspire new generation of scientists in plant biomechanics. With that, it is my great pleasure to officially open webinar series 2025, Road to the 11 International Plant Biomechanics Conference 2026. Thank you very much, and I wish you an engaging and fruitful discussion. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much for delivering the remarks, Dr. Sigit Sunarta, and also for your continued support to our upcoming event, the 11th International Plant Biomechanics Conference 2026. And now we're on to the main event. Today's event features three distinguished speakers: Professor Dr. Joseph Grill from CNRS France, Dr. Yu Chen Long from the National University of Singapore, and Dr. Jasmara Master of Agriculture from Universitas Gadjah Mada. And we are pleased to be joined by our moderator, Dr. Hairi Cipta, who will guide us through the presentations and discussions. I would like to shortly introduce our moderator for today. Dr. Hairi Cipta is an assistant professor in the Faculty of Forestry Universitas Gadjah Mada, and his research focus is about wood anatomy, X-ray CT imaging, image analysis, and related fields. His latest publication is about stem graphic tropism, tension wood formation in 3 tropical woody species with different wood densities. And now I will hand over this session to our moderator, Dr. Hairi Cipta. The floor is yours.
Okay. Thank you very much, Dr. Sidianifa, for the introduction. Okay, good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. I hope you are doing well. Whether you are joining from Yogyakarta, other parts of Indonesia, and also from all over the world. It's good to see familiar names and face, and also some new face in this today webinar. Distinguished Dean of Faculty of Forestry, honorable speakers, and also organizing committee members and all participants. Let me introduce myself. My name is Hairi Cipta from Faculty of Forestry, Universitas Gadjah Mada, Yogyakarta, Indonesia. So today I will be moderating this scientific session. Okay. Before we begin, let me thank everyone for taking the time to join this session. You will find it both informative and also inspiring. Okay, with your kind permission, allow me to get to this discussion. So introduce our distinguished speaker. So we have 3 experts joining us today. First, Professor Joseph Grill, who will present introduction to plant biomechanics. Hello, Professor.
Hello, thank you very much.
And okay, the second one, Dr. Yu Chen Long, who will discuss about the mechanohydraulic growth of plant cells. He's from the University of the National University of Singapore. Hello, Dr. Long.
Hi, everyone.
Hello, everyone. Hello. And the last speaker is Mr. Rafi Pujasmara, who will share about growth stress with properties relationship in Cyclone 20 in Yogyakarta. Hello, Mr. Rafif.
Hello, good afternoon from Nagoya.
Okay, hello. Okay, together they will take us on a journey from the cellular to the structural level of plant biomechanics, how plant grow, respond, and adapt mechanically to their environment. Okay, first, let's begin with our first speaker. Let me introduce his CV. So, Professor Dr. Josel, he is from France, is a senior researcher at CNRS. So currently, I think he's in the two institution now, Institute Pascal and also in PIAF in the Clermont-Ferrand. So he earned his engineering degree from École Polytechnique and also the EA from Pierre and Marie Curie University, and also his PhD in the same university. He has some experience like visiting professor at Kyoto University and also adjunct professor at University of Tehran. So please join me in welcoming Professor Dr. Joseph Grill. Let's give him applause. So Professor Grill, this is your time to present your presentation.
Yes, hello. I will try to find my presentation. Yes, here.
Okay.
Can you
yeah.
see my presentation?
Yes.
Yes.
So, thank you very much, Professor Heidi Sipka. It is for your introduction. I am very pleased to give this introductory lecture to the webinar. So I will focus on the growth stress in wood stems, which had already been introduced last time by my Japanese colleagues. And I will complement their presentation by talking a bit about the setting up of growth stress. And if enough time, I will talk also on about the rheology of wood formation. As was previously said, I am now in Clermont-Ferrand. I share my time between two labs, one Pascal Institute for Wood Mechanics and PIAF for tree biomechanics. And the research I'm going to present now is what I did before coming to Clermont-Ferrand in Montpellier when I was in LMGC. Before starting, I give just very few words about the research that is going on in PIAF on tree biomechanics, which deals with different aspects of wood mechanics, especially. So the question investigated in the MECA group of PIAF is how the trees stand up and grow in a changing environment. So, which means, well, that's in the context of climatic change, when you have lower daily winds and higher frequency of extremely windy events. There are really two aspects behind that. One is the acclimation, such as the acclimation to daily winds and the resistance of trees to breaking. And these leads to the study of the growth mechanoregulation. And there is another one, which is the resilience of trees after strong events, which leads to the control of posture, the interaction between graviperception, photoperception, and proprioception. So now coming to my subject about the stress patterns in growing stems, I will first mention the concept of primary and secondary stem growth, which is, what you have in any living system, any plant or any living body, actually, any living body grows according to the rhythm of primary and secondary growth. Primary growth is occupation and space, and the secondary is the reinforcement in that space. And so concerning the woody stems, you have the deposition of growth units, which concerns the primary growth of that stem, and the deposition of wood layer around the piece until the cambium, division of cells in the cambium and deposition of wood layers, which is the secondary growth. And I'm mostly interested in what happens in the wood, which is between the pith and the cambium. And what I'm going to show in the few slides now is to explain how the stress pattern in a tree stem results from the balance between primary and secondary growth. So we are sorry, I should remove that. I don't know how to remove that part. Oh, yes. Okay. So we want to study the growth stress, and the stress will be written in this presentation as sigma. It is the stress supported by the wood at position X along the time t. And here we focus on the axial or longitudinal component of the stress, which will be positive when it is a tension and negative when it is a compression. So the transient phenomena like wind and snow are very important for the tree growth as a signal, but they do not have any direct effect on the growth stress. Only what is permanent and is occurring at the same time as growth is important for this calculation. So we have first the gravity, so the dead weight of the parts of the tree supported by the wood, and the stress, what we call the maturation stress at the periphery of the stem. So we are going to explain later. So the calculation is based on two principles. First, we have additivity of the stress increments. This is called the superposition principle. And then the second one is that the wood is only loaded from the moment it is formed. This is quite obvious, but it makes all the difference between conventional mechanical calculations and calculations applied to growing bodies. So since we deal with slender structures like branches or stems, we apply the beam theory, but only in an incremental way. So we take into account the increase of size and the increase in loading. So first we start with the effect of gravity. So if you consider a section of the cross section of the tree, and where you have wood, you can divide the effect of the crown on that part of the trunk into two components. One is the axial component, which is mostly a compression, and the other one is a bending component, which is due to the orthogonal component of the stress of the load. Sorry. So according to the superposition principle, it can be divided in two parts. One is the axial component only, what we have in a purely vertical stem, and what in horizontal, and the bending component, which is what we have in a purely horizontal stem. So now first take the first case. If you have a well-balanced trunk and you want to study the effect of gravity, if you had an artificial pillar that was made, fabricated and then loaded, you would have an homogeneous stress compression at the bottom of that pillar, which can be calculated as the weight divided per surface, which is the density times the gravity times the height. Now, if you consider the same situation but with a growing body, and you want to reach the same end final situation, you start, we apply the second principle. It must exist before it is loaded. So only at the beginning, only the central part is loaded. And then when you have an additional weight, it is loaded again, but with the load divided along the current surface, and again at the later stages. And in the end, the stress distribution will be very different from what it was in an artificial column. And this can be calculated mathematically. And what happens, what can be observed is that it is very dependent on what we call the allometry, which is the relationship between the primary and the secondary growth, which can be expressed by the relationship, like a power law relationship between the height and the radius with the power n. And you can have such a formula to describe that. This stress, the shape of that curve will be very dependent on the parameter n. Like if it is 2/3, which is quite common in trees, it will be that expression, for instance, which is shown here. Just to say that the level of this compressive stress is usually very small. If I take an extreme key case of giant trees, we only have a maximum stress of 4 megapascal, which is very small for wood. Now the effect of gravity for a horizontal stem. In the same way, if we take an artificial beam anchored at one end, if it is subject to gravity after being made, it will have a tension on the upper side and a compression on the lower side in a linear relationship. Now, and then it will deform a bit because of that. You have a bending curvature occurring because of that moment. Then, if you do the same, and it can be easily calculated according to beam theory, which is a proportionality between the stress and the transverse direction and the vertical direction. With the m is the moment and I is the moment of inertia. The m is the bending moment and I is the inertia. And if you do the same with the growing stem, horizontal stem, you would have first a small bending, then additional weight and a bigger bending, and then more weight and more bending. And in the end, you have a superposition and you have a very different stress distribution than what you have in the standard case. And again, the shape of that curve here will be very much dependent on the allometry, which is like in 2/3 according to such an expression, but this can be computed for any value of n. The amount of stress here is much higher than what you have in vertical trunks. So this stress is not negligible at all. And now going to the maturation stress, which is what we have at tree periphery after wood is formed. So you have a new layer of wood which is deposited at the periphery at each, in Europe that is each year. In tropical countries it can be according to a different rhythm. When the wood cells are made, after the wood cells are made, they tend to contract actually and expand transversally, so that because they are attached to the previously made trunk or branch, it will be under axial tension and transverse compression. Now let's consider the case of a well-balanced stem. Doesn't matter that it is vertical or horizontal, but we imagine it vertical. If you have tension in the first inner layer, and you would have compression in the inner layers to balance the tension in the outer layer.
Sorry, Professor, you have 2 minute lapse.
Sorry?
2 minute lapse. Sorry.
2 minute lapse. But 5, no? Okay, 2.
Okay, 2, yes.
Okay. Then I have additional tension, and then I will have this kind of pattern, which is known as the Kubler model. With a high tension. So the level of tension we have here is about half, is the same order of magnitude as the elastic limit, so it is very high. Now, if I want to, thanks to that, you can adjust the maturation, you can adjust the, if you have a branch that goes in a direction, wants to go in a direction, sorry, if you have a stem that wants to go in a certain direction to the right here, it can obtain that by having more wood made on one side, and then you would have, in the end, a bending through the right, to the right. And then you can have the same effect by maturation asymmetry, like in tension wood of hardwoods, you have higher stress on one side, and the same you can have a movement to the right. And then you can also have the strategy of softwoods with compression wood, and then you have negative stress on one side and positive stress on the other. And then you obtain also very different stress distributions. And then if you consider a horizontal stem, you can have the stem can be kept horizontal by the maturation stress. So you have a tendency to bend, which is counteracted by the maturation stress, and you can adjust the stress that at each time step, and then in the end, the stress distribution will be a positive stress on the upper side and a negative stress on the other side. So you can see that you have very unconventional stress distribution due to the addition of stresses. So I give here a short summary. So the idea is that the mechanical actions undergone by the wood of the tree are additive, so that you can study them separately. If they are permanent enough to accompany this growth, gravity, and maturation, we speak of growth stress. By applying the strengths of material theory at each stage of growth, the spatial distribution and temporal evolution can be computed for a few basic cases, and then they can be added to each other to represent any situation. So you have the effect of gravity or the cell wall maturation. You have the vertical stem or horizontal stem. You can have the eccentricity induced curvature or maturation differential induced curvature, which is reaction wood. And you can in this way model such things like the stationary growth of inclined stems. So I think if I only have no time left, I stop here.
Yeah.
Okay.
Yeah.
Okay, I can stop here.
Okay. Thank you, Professor Grill, for the wonderful presentation. Okay. Sorry for very. And so if Professor Grill already explained to us about the growth stress on the living trees, and if participants have any question, please keep them for the end of the session for the Q&A session. Okay. Give applause to Professor Grill. And we move on to the next speaker. So I would like to introduce him. So Dr. Lee Cheng Long is assistant professor at the Department of Biological Sciences at the National University of Singapore. His research combines plant developmental biology, biomechanics, tissue hydraulics, and also quantitative plant biology. He completed his PhD at Utrecht University in the Netherlands. He also worked before joining NUS. He also worked as postdoctoral researcher at Lyon, research scientist at INRAE Lyon. Today, he will present about the mechanohydraulic growth of plant cell. Please welcome Dr. Chang. Screen is yours, and you have 15 minutes.
Okay, great. Thank you very much for the introduction, and also thank you for the invitation to this webinar series. Yeah, so my name is Yu-Chen Long, and today I'm going to talk about the mechanohydraulic growth of the plant cell. I think if you have kept house plants before, you must have seen something like this. If we forget to water the plants, then the plants might get very soft and will be droopy towards the gravity. But if we rewater the plants, then the plants may actually accumulate internal pressure so high that it allows the plants to prop up their leaves and then resist gravity. Right. So this behavior is actually giving thanks to the existence of a very special structure in the plants, but not in animals, called the cell wall. Right. And I think this textbook image shows clearly that without a cell wall, when animal cells are dumped into a hypotonic condition, water will enter the cell through plasmolysis, and without the strong cell wall, the cell may actually burst. But with a very strong cell wall, when water enters the cell, actually the cell wall can sustain a much stronger mechanical stress, therefore actually blowing up just like a balloon in an analogy, right? And this water uptake is driven by osmosis, and I think for this audience we don't really need to introduce too much about this, but just to have a short reminder. So osmosis happens when the cell have a higher osmotic pressure than the extracellular space, which will drive a osmotic intake of the water, indicated by this J osmotic. Right, the water influx actually accumulates inside the cell and generates a hydrostatic pressure, or called turgor pressure in the field of a plant and other wall organisms. And this turgor pressure actually pushes the water out, as indicated by this backward arrow and indicated as J hydrostatic. Right, so this water intake and output keeps on going until the level of the turgor pressure reaches the same level as the osmotic pressure, which in this case, the J inward and the J outward becomes equalized. So in this case, we have a net water flux of 0. Right, this mechanical pressure not only pushes on the water, this turgor pressure does not only push on the water, but it also actually push on the cell wall and stretching the cell wall so that it can accumulate a tensile stress that can also actually pull the cell wall outward. Now, in woody structures, the cell wall can be very, very strong, I think, as introduced previously by Joseph. But in plant tissues where the cell wall is softer, especially in growing tissue where the cell wall is primarily in the primary cell wall structure, then the cell wall is not only resisting the stress, but also can have a pliable property. So as indicated by this molecular simulation, the primary cell wall is composed of major polysaccharide components like cellulose microfibrils or zelloglucan or pectin, and that are all embedded in a water-rich environment. In other words, the primary cell wall can be considered as a fibrous network that is embedded in the hydrogel. And because of this nature, it has been found in many different experimental systems that the primary cell wall can both resist mechanical stress, but also can elongate when the stress is applied to it. So as shown here in onion epidermal strips or in Arabidopsis leaf epidermal strips in some of the recent studies, can see by repeated mechanical loads, the cell wall fragment can actually go through different phases of reversible and irreversible expansion. So indicating that actually cell wall is not only just elastically restricting the pressure and the tension, but also can actually slowly creep, thereby contributing to the growth of the plant cell. Right, and this growth on the microscopic level has been subjected to, for example, the sliding of the microfibrils inside the cell wall. And this sliding can also contribute to these multi-scaled cell wall mechanics-based principle of plant growth and morphogenesis, which ramps from the molecular scale all the way up to organ or even organismal scale. So this process has been very nicely reviewed by Rico Cohen and Daniel Costco very recently. And in their recent publication, they also have two interesting sentences that I actually want to quote here. So this microfibril sliding can actually dissipate cell wall stress. And this cell wall stress relaxation actually also generates a slight water potential disequilibrium that is required for the cell to keep on taking up the water during the cell enlargement. So these two statements actually are very interesting, and it tells us that the cell wall stress, the water equilibrium, as well as the cell expansion are somehow coupled in this process. And indeed, this coupling of cell wall stress, turbo pressure, as well as cell expansion has already been summarized and modeled by Lockhart and Ortega for more than 20, or actually 40 years ago. And this process can be summarized by this illustration over here. For example, mechanically speaking, the primary cell wall, when under turbo pressure from the inside, will start to accumulate a cell wall tensile stress on the outside, as indicated by the sigma over here. When pressure increases, this can result into, for example, by water uptake, this will result into a cell that's slightly expanding. But if this pressure is removed, then the cell wall may actually shrink back. This indicates that the cell wall can actually have an elastic behavior. But if the cell is allowed to be inflated for a much longer period, and also this pressure inside is higher than a certain threshold, then the cell wall may also undergo a irreversible expansion that is both plastic and may also have a viscous behavior. And in this process, the cell wall actually will expand and creates extra space in the cell. That is what we call cell expansion. So as said, this process of the extra cell wall area creates extra space. That means the space needs to be filled up by certain substances. And for plant cells, that is mostly filled up by the uptake of water, which is driven by osmosis. So it happens that when turbo pressure is smaller than the osmotic pressure, then there will be a net water uptake. And then when pressure reaches the osmotic pressure, then there will be a net zero water flux. So you can see here, turbo pressure actually unites or actually couples the mechanical and the hydraulic aspects of the cell expansion. And this has been summarized in the Lockhart-Ortega model. I think this is not unfamiliar for many of the plant biologists over here, but I think it's also very nice to notice that the notion of mechanohydraulic coupling has also been picked up by scientists actually in different fields of biomechanics. Here, I list out a few recent examples on how animal systems may also be regulated by this mechanohydraulic coupling, where both lumen genesis and as well as tumor growth or even cell migration may be under control of the mechanohydraulic coupling. And I think this realization of the importance of this coupling really excites me. Yeah, but anyways, back to our plant study. So as we have already explained, so I think I will not go into the details of the one-dimensional Lockhart-Ortega equations, but the idea here is that the original model actually models the elongation or the one-dimensional growth of a plant cell, where for the cell wall elongation, the cell wall relative growth is controlled both by the reversible and the irreversible elongation of the wall. Right. The water influx actually is determined by both the sum of all the influx and the efflux of the water, as dictated by the water conductivity of the membrane. Now, previously, when I was in the lab of Araski Budaud in Lyon, we collaborated with the lab of Christophe Godin and Ibrahim Shadadi, who actually came up with this two-dimensional extension of a vertex model, where Ibrahim modeled the cell wall rheology of the Lockhart-Ortega equation, but instead of giving the model a turbo pressure that initiates the growth, as previously many people do, he actually allowed water to be dynamically taken up by osmosis, and also allowed the water to flow between the cells through plasmodesmata, as indicated by these blue arrows over here. By combining this dynamic hydraulic control with the mechanical balance and the cell wall rheology, he actually predicted that the cells may have different pressures in different tissues, actually within the same tissue. And this prediction has been validated by our atomic force microscopy, and this work has been published, so I will not go into the details of that. You're all welcome to read our work by Ibrahim and also by myself. But then we ask the question, like, how actually this mechano-hydraulic coupling regulates growth, and more specifically in the context of a three-dimensional cell growth. To study that, we first imagined a very simple cell geometry, namely a spherical cell that also have the same Lockhart cell wall rheology. But when we talk about a three-dimensional cell, we see that a term called surface to volume ratio starts to emerge. And that is, when you have when you calculate the surface and the volume, you see that actually the surface actually scale to the volume with the reciprocal of the radius. That means when the cell is larger, when the radius is larger, we actually have relatively less surface per volume of the cell. And this limitation in the three-dimensional geometry is actually very interesting because when we extend that to the cell wall elongation, we see that now actually cell radius become part of the cell expansion of the mechanical aspect. And what it tells us is that when cell radius increases, actually cell wall has less and less resisting power over cell expansion. Therefore, the cell will grow faster and faster, as indicated by this green curve over here. For the hydraulic limit, we can also understand that when the cell gets larger, then there's less and less surface area to conduct water influx. Therefore, when the radius is larger, cell actually grows slower, as indicated by this blue curve over here. Now, because the cell has a mechano-hydraulic coupling, therefore, by having the two limits, we actually predict a new growth curve where the cell actually have a first faster, then later slower non-monotonic expression cell expansion rate that is not regulated by any of the mechanical or hydraulical parameters. So it tells us cell geometry alone is already possible to explain actually the growth onset and the growth arrest of a plant cell. We further extended the analysis of the model by actually extending them to more arbitrary geometry, and also we found, in short, that actually a geometrical parameter f that is able to combine the geometrical, the anisotropy, as well as the neighbor number of the cells, so the tissue context, therefore actually greatly extending the applicability of our model. Right. To test the feasibility of the model, our postdoc Kartik Babu actually utilized a very special plant material. He used the shoe-typical meristem of the Arabidopsis plant and treated with a drug called orizolin, which prevents the microtubule polymerization. So this removes any microtubule-mediated mechanical feedback and also stops the cell from dividing, so the cell can keep on expanding without any division, allowing us to follow the expansion very nicely. We also think that the cell actually loses part of the mechanical feedback because some of the cell can keep on expanding so large until they explode, as indicated by these little stars over here. But even with this lack of the microtubule-mediated control, we can see that the three-dimensional growth rate over here is actually showing a faster than slower trend, indicating that cell size may indeed have a function on the cell side on the growth regulation. And when Kartik actually analyzed over 260 cells over, I think, 5 different days, and then he found that actually all these growth trajectories collapse onto a curve that is very reminiscent of what we have predicted, where when the cell gets larger, the growth rate may first speed up and then slow down. But of course, the mechanical property is likely not constant over time, and there can be some scenarios where, for example, when a cell gets larger, the cell content might be diluted. And if that is the case, then it means that the osmotic pressure may actually follow, may be proportional to the radius to the minus of power 3. But if there is osmotic pressure homeostasis, meaning that osmolytes are being actively pumped into the cells, then what we may also have is a constant osmotic pressure that is proportional to, let's say, the radius of the power of 0. So here, Kartik measured the osmotic pressure of the tissue in different times by incipient plasmolysis. And what he found is that actually the osmotic pressure follows the power of a minus 0.8. That indicates that the cell is actually actively pumping in osmolyte, but it's not being able to keep up with the expansion of the cell. Similarly, we also know that the cell wall actually thickens over time, and quite surprisingly, our target also found that the cell wall actually softens over time with the exponent of also surprisingly minus 0.8 by coincidence. So this suggests to us that actually the mechanical property and hydraulic properties are highly dynamic. So we included these dynamic control of these parameters and derived the new model, which have actually a power law behavior both on the mechanical and hydraulic branches. We used this model to fit the growth data and actually found that it seems that the total mechanical dynamics and the total hydraulic dynamics can have actually different regulatory powers. Now, this indicates that maybe other parameters like the water permeability, cell wall extensibility are also differentially regulated over time. And this is something that we're following up right now. But with that said, I want to make the conclusion. So we first identified a cell size autoregulation that is imposed by cell wall mechanics, tissue hydraulics, and cell geometry. Quantitative three-dimensional imaging pipeline experimentally confirmed the prediction of this non-monotone growth trend. And direct physical measurements reveal the cellular mechanics and hydraulic of the biomechanical properties are highly dynamic. And this modeling framework may actually recapitulate the many of the sigmoidal growth trajectories that are found in various biological systems. And with this, I want to give thanks to the Kartik Babu, who's over here, who did all the work, and also all our lab members and collaborators in the funding agencies. And with that, thank you very much for your attention.
Yeah, thank you, Dr. Long, for your presentation. Okay, you have a very interesting presentation about plant mechanohydraulic in the cell level. Do you have time for discussion, Dr. Long?
Yeah, maybe some minutes. Yes.
Okay, maybe one or two question. One question maybe. Yeah, if any participant have one question only for Dr. Long, please raise your hand. If you have any question. Okay, Mr. Lindsay. Mr. or Miss Lindsay?
Hello.
Oh, yeah. Miss Lindsay. Okay, please.
Okay. Yeah, great talk. I like it very much. So I wonder, we talked about cell wall softening during the mechanical sensing and osmolarity and hydrophilics process. And I wondered what is a plasmodesmata could play in between the cell. So is there any parameters or any observations previously has been indicated that plasmodesmata is somehow play an effect on this model's establishment? So this was basically my question. Thank you.
Thank you very much for the question. Short answer. So I think first of all, plasmodesmata is indeed a very important parameter that may actually drive the so-called hydraulic backflux of the water. Where the cell can take up water, but then the pressure can actually squeeze them out into the neighboring cells. So indeed, in a multicellular context, actually, if I may see if I can find it. So previously, actually, Ibrahim's model included plasmodesmata as a important parameter. And what he found is that actually plasmodesmata, I'm actually trying to find the slide, but I don't think I see it. So anyways, maybe I just go back to the original slide. Sorry. That is over here. Yeah. So actually plasmodesmata will actually indicate this LS parameter. And it was found that actually the balance between LS and the LA, which is the plasma membrane osmotic water uptake, actually controls the behavior of the model. So that shows a very important role. Now, in our current three-dimensional model, actually, we are only still working with single cells. And although the geometrical analysis can account for partly the tissue context, we believe actually the water permeability itself can also serve as a combination of the water uptake as well as the plasmodesmatal water loss in this process. So it is partially involved in the new model, but I think more analysis is definitely needed. And to answer the question maybe about the plasmodesmata and the cell wall softening, this is something that we don't know. But maybe indeed the plasmodesmata and the cell wall composition over there may have a global contribution to the cell wall softness as well. This is something we don't know.
Okay, good. Thank you very much.
Thank you.
Thank you so much.
Okay, thank you, Miss Lindsay. Maybe one question, Dr. Long?
Yeah, I think it's okay. Yeah, I think
Okay,
I just
okay.
need to go in 2 minutes.
From Gabriela Mosca. Okay, sorry.
Hello, thank you.
Okay, yes.
Thank you for the very nice talk. I'm curious to know whether in your calculation of how the cell gives in into growth, and you estimated sort of power though, depending on already how much the radius is increasing. And I can't remember the slide number. If that is also considering the mechanical stress that is accumulated in the cell wall, which would, I think, fit also with what you are saying.
Yes, indeed. Yeah. So I think I rushed a little bit over there on the explanation here. So indeed, actually, for this branch, the power law actually is a sum of all the dynamics of, let me see on the whiteboard, actually the cell wall extensibility, cell wall thickness, cell wall threshold, cell wall Young's modulus, as well as a geometrical parameter for the mechanical aspect. Yeah. So indeed, this N summarize all of that. And experimentally, we see some of these parameters already start to show up. So what we think the predictive power over here is that by fitting this power law and then actually determining some of the power, the components, we may also predict the other behaviors of the mechanical properties that are difficult to measure. And we are hoping to actually use this to also study how the stress can actually be relaxed in this scenario as well. Yeah, not sure if that answers your question.
Yes, and I think then it fits like if you connect to these publications connected to the largest empty circle in a growing cell and the stresses that depend on the growth along 2 dimensions. I think it fits.
Yeah, exactly.
Fit with what you are showing there. Yeah. So well, I mean, very good. I'm excited to see how this all then turns out.
Okay, well, thank you very much, Gabriela.
Thank you.
Yeah. Okay, sorry, everyone. I think I need to run for my next lecture, which is starting in 3 minutes.
Okay, thank you, Dr. Long. Bye-bye.
See you.
Yeah, see you. Okay, sorry, because Dr. Long have a class today, so he have to leave the room earlier. Okay, we move to the last speaker, the third speaker, Mr. Raffi Pujasmara. So let me introduce Mr. Raffi. So he is currently assistant professor at the Faculty of Forestry, Gajah Mada. So his research focus on wood physics, wood anatomy, and also the basic properties of tropical wood species. So with special interest in growth and its impact on the wood performance. Actually, he is my colleague in this faculty, and he completed his bachelor's and master degree in business Gajah Mada, with part of his research conducted at Kyoto University, Japan. Now he is currently in Japan pursuing his doctoral studies at Nagoya University, Japan. And today he will present about growth stress with properties relationship in a town planted in Yogyakarta. Please welcome Mr. Pujasmara. Screen is yours.
Okay, thank you, Dr. Hairi, for the time and opportunity. Let me share my screen, and I hope the sound is okay. Let me hide some of the images here. Okay.
Yeah, okay. The sound again. Yeah. Your slide are also okay.
Okay. Okay. Thank you again. I'm very honored to join this webinar, but actually, I just happen to be one of the staff at the Faculty of Forestry, UGM. So I'm very nervous since I am a newbie among all the other speakers. I believe I have more to learn today, but I will try my best to present. And before I start, as a part of the Faculty of Forestry, we hope to see you in the BBM conference next year in Yogyakarta. You can check the website as seen below in this image. So today I'll talk about our previous research on the growth stress and wood properties in teak grown in Yogyakarta. Let me begin. I believe you are already familiar with teak. It is a valuable wood species, and today we are going to discuss about one of the thick clone or thick variants. I call it Jati Biotrop. Actually, it's to make it easier because the producer is from Biotrop. It is produced from tissue culture, and it is claimed to grow faster, have 3 times yield than conventional thick, and can be harvested at 15 to 20 years old. And the problem is people usually look down to the fast-growing ticks because they see it as lower in quality. The reason is uh, uh, juvenile juvenile wood that is bigger portion in uh, fast-growing ticks because it cut at a young age, and also defects that caused by or related to growth stress that are common in some of the tropical wood species. One of them is thick. But the question is the quality of this material or the Jati Biotrop is really poor, or we just don't have the necessary information? That's why we conducted a research to observe the growth stress level and the variation in wood properties, and we tried to link the relationship among these two factors in this Jati Biotrop grown in Yogyakarta. So for the study, we took 3 trees at 10 years old with average height of 17 meters and average diameter of 17 centimeters. It is planted in near the south coast of the Java Islands. And the basic activities of this research is first, we are measuring release strain using the strain gauge method. We measure the longitudinal SRS and the tangential SRS also. We took it in the 4 cardinal direction, north, east, south, and west, respectively. And then after that, we also measure the internal or residual strains with the plank made from the trees. And basically, we place the strain gauge to show the difference in the positions like a near bark or mid radius, near pith, center from north to south directions. And then we took specimen from near the measurement points to observe the wood properties such as moisture content, density, fiber length, the MFA, the MOE, then some chemical contents. And then we analyze the relationship between those first and two. So for the results, first I'm going to show you the surface release strains. So the longitudinal SRS is range from minus 1,200 to plus 300 microstrains, and the tangential SRS is range from minus 780 to plus 380 microstrains. The majority of the longitudinal SRS were negative, showing the reduced dimension due to the release of tensile stress, and the majority of the SRS were positive, showing the increased dimension due to the release of the compression stress. So the majority, we have some values in tree number 1 that is not belong in the dominant pattern. But if we set that aside for a while for the tree number 1, the SRS was predominantly less than 500 microstrain. And if we compare that to the other thick, like a community forest in the nearby area or the conventional thick, the SRS here is lower. So we can see the SRS was low. And if this was a result of tree breeding or genetic selection, this is a very good start to obtain less defect material with selecting a thick with a lower growth stress level. Oh, sorry. And now I'm going to talk a little about what happened to tree number 1. Actually, we didn't make a clarification analysis, but we tried to find some of the hypotheses from the reference, and we find 3 possibilities. One is maybe the occurrence of tension wood. As you know, in the last webinar, we heard about the tension wood that grows in the straight stem. Maybe this was also there are occurrence of tension wood because the threshold is already surpassed. And the second possibility is the juvenility, because young trees with a significant amount of juvenile wood maybe exhibit different patterns of growth stress. And the third, maybe because of the location is near the coast, maybe the moisture in the soil is different, or maybe the winds are making something, because a similar phenomenon was also reported by some researchers. So that's for the surface release strains, and I'm going to continue to the internal or the residual strains. This is the longitudinal one. As you can see, the values on the outer side were negative, showing the reduced dimension, and the center part is positive. This is likely a normal pattern. And if you can see the range of the strains here, from minus 580 to plus 790 microstrains, it is likely smaller than community forestic in nearby areas where the comparison is much higher in negative and much higher in the positive strains. So if this also because of the genetic selection, I think this is a good result. But anyway, why I say that this is a low strain, because we didn't find no immediate defect after cutting. However, sometimes later, some splitting occurred in the remaining specimen wood saved in the log yard of the plantation and in the storage room. I thought maybe this is attributed to the later release of the residual stress. In this case, I think we have to find some method to handle the pre-harvesting and the post-harvesting to get the material safe, not from defect. Okay, so the release strains already help us to understand how the wood behave after cutting, and to deepen our understanding, let's take a look at the wood properties and how they connect with the growth stress by the correlation with the strain. First, I'm showing you the table here, variation of wood properties of 10-year-old Jati Biotrop, and this highlighted parameters are the ones who have a significant difference in the radial directions. So there is a fiber length, the MOE, and the lignin. So I'm focusing on that because of our limited time. And the second one is I'm showing you the table of the correlation with the internal or residual strain with the wood properties. And you can see there is a correlation between the MFA, the fiber length, the MOE, the lignin content, and the hemicellulose content. Okay, I'm skipping the other part. Now let's see from the microfibril angle. As you can see, the microfibril angle in the center was about 21 degree, and the outer part was 12 degrees. And this is likely a normal pattern where in juvenile wood are higher, and as the wood get a lateral growth, the MFA got lower. And this result is similar to Bhatt et al.'s report, which he made a model in from thick in India. And I think maybe this one to be compared is still in the juvenile phase. So this tree is still in juvenile phase. And if we compare the MFA here with Jati Biotrop planted in other place, this MFA is lower. Yeah, the lower MFA in one side is good because the dimension is more stable. Sorry, but lower MFA is also associated with the wood with high tensile stress, and we have the correlation here. So the section with the low microfibril angle experiencing higher tensile stress. So maybe this tensile stress in microfibrillae regulates the magnitude of the growth stress, and because of the correlation here, we have to know if the low MFA mainly it reflects the normal tree growth, or it actually correlates more with the higher growth stress level. So low MFA may improve stability of dimension, but we should also be careful about the high tensile stress. Now to fiber length. In addition to the original 3 radial position, I measured per 1 centimeter intervals. As you can see, the values here showing a upward trend from pith to bark. And the statistic confirms that all 3 radial position, the near pith, middle, and near bark, is significantly different, which indicates also the xylem maturation. And the value is also consistent with Bud's model. So I suppose this tree is still on the juvenile phase. And generally, longer fiber are preferred because they are associated with flexural strength, which is proven in the correlation with MOE. But we need to be cautious again because section with a longer fiber length experiencing higher tensile stress. This is a similar report by previous research, and I think future research must search whether this correlation is dominantly because of the growth stress, or this is just a normal phenomenon because of the lateral growth. And we continue to the MOE. The average MOE here is 11.2 gigapascals, and if we compare to another fast-growing ticks, this is quite high, and maybe even with conventional tick, this is higher. And so this is good result. By for utilization, we have to be careful because this have a different MOE in the inside part and the outside part. So for small diameter, we need to be careful because this tree is only 17 centimeters in diameter. And also, the correlation with the internal stress, we also have to be caution once again, cautious. Section with higher MOE experiencing larger tensile stress. So, is this correlation mainly affecting the properties of the wood? It's still a question for me. And lastly, in the last properties, we say this about the chemical composition. We have the lignin with 23.5%, cellulose with 38%, and hemicellulose with 34%. And actually, lignin and hemicellulose here is still fell in the usual range, but the cellulose is lower than this usual reference book we usually use. But compared with other jati biotrop planted in other areas, it is still comparable. The values here. Maybe it's just because of the difference of the planting site. And for the correlation with the residual strain, we found a correlation with lignin and hemicellulose with residual strains. This is actually like opposite of what I usually read. Usually in hardwoods, large tensile stress is often associated with high cellulose content and low MFA, according to the cellulose tension theory, while in softwood, large compressive stress is often associated with a high lignin content. But we don't have a correlation with cellulose here. It's the lignin content. However, I think I found some reference that each theory alone is not sufficient to explain the phenomenon, and there is also a unified hypothesis that suggesting that both cellulose tension and lignin swelling mechanism are required. So I don't know, maybe this is still inside theory. But some suggestion why didn't find the correlation with cellulose. First, it's because of the limited number of samples, of course, that maybe it can forming an unpredicted correlation, and also there is a limitation on the method applied by for the determination of chemical content, as it mentioned by referred paper that I refer the method to, and there is no significant difference in cellulose content among position that I observed. That's why maybe the correlation didn't show up. So let me briefly summarize. The first, the Jati biotrop tick showed moderate growth stress level. Why I say this? It's because compared to the previous reports in tick, and because of the no immediate defects after cutting. Usually we find some immediate defect after cutting ticks. And this is highlighting the importance of the tree breeding or tree selection for lower stress level. But for the later defects that still occurred, it invokes us to find a correct method or correct treatment in the pre or post harvesting the tick. The second one, these Jati biotop tick properties were comparable to the other fast growing tick variants. Some radial variation in wood properties were found, therefore utilization must be done cautiously for the small diameter trees. And from fiber length and MFA, it seems that the trees were still on juvenile phase at 10-year-old. So we have to use it very with caution. So, and then the third summary is no clear relationship with SRS. So this is an experience for me. So I need more measurement points in the future. But how much is the minimum necessary? Because sometimes we cannot cut the tree that much. Maybe some plantation only allow us to cut a very minimal number of trees. And because IRS correlated strongly with MFA, of fiber length, modulus of elasticity, lignin, and hemicellulose, some were explainable, some still in question for me. And this finding, but at least we have some key factors to observe to manage the growth stress level in the fast-growing teak plantation. I think that's all for me. Thank you and terima kasih. I return this session to the moderator.
Okay, thank you, Mr. Rafif. Okay, applause to Mr. Rafif for the nice presentation. Okay, before we move to the Q&A session, I would like to summarize the three presentation. So the first presentation from Professor Joseph Kriel is about growth stress occurs when mechanical forces in wood become permanent during the growth. And it can be analyzed using strength of the materials theory for different stem conditions like vertical, horizontal, curve, or inclined. From the second speaker, from Dr. Yu Cheng Long, cell growth is controlled by cell wall mechanics, tissue hydraulics, and also geometry. 3D imaging confirms nonlinear growth patterns, showing that biophysical properties are dynamic. And imaging modeling framework also helps explain the typical sigmoidal laws of biological systems. And from the last speaker, Mr. Rafif, who present about the biotrop or tick, moderate growth stress with no immediate effects. Emphasize also the need for proper selection and treatment. Its wood properties were similar to other fast-growing tick, but still juvenile at 10 years old. Okay, we have maybe 20 minutes for question and answer session. If you have question, please raise your hand and we will unmute you. Or you can type your question in the chat box. Okay, please raise your hand if you have a question to Professor Joseph Kriel or Mr. Raffi Pujasmara. Okay, maybe we have
There is
some
one question in the chat box.
Yeah, yeah. Okay. So okay, for Professor Bill, can the plant biomechanics describe also have an impact on the generative process of trees and be used for similar studies on non-timber forest products such as nutmeg, herbs, or cinnamon? Okay. Maybe you can answer the question, Professor Bill.
Yes, thank you for the question. Yes, yes, this. I'm not sure I understood the first part of the questions. Yes, what I did concerns the process of tree formations. What is missing in this model is the signal that provokes the growth. So this has to be coupled with a model that takes into account the various signals that can induce the growth. But if you do that, then you can have a description of the variation of the development of the trees. But this can be through modeling approaches, and that is possible, yes. But concerning the non-timber forest products, what I described only applies to the situation where you have addition of material. So, well, in the case of non-timber products, you may have well, I don't know how it works with cloves or cinnamon, but you certainly have addition of material, and the progressive addition of material and the progressive loading always produces, in theory, the similar situations.
Okay, thank you, Professor Bill. Mr. Nurfianto, do you have any follow-up question or enough? Okay, thank you. And the next question from Miss Yulian. She has question to Mr. Rafif. Why did you choose Jati Biotrop for your research? Why is it Jati Biotrop? Please, Mr. Rafif.
Okay, thank you, Ibu Yulianti, for the question. Why I choose Jati Biotrop is actually because of the fast-growing plantation is fast-growing teak plantation is now like everywhere. One of the location for the plantation is in Yogyakarta. It is interesting because the Jati Biotrop it's made from a tissue culture, and the claim was quite great, like 15 years to be harvested for a teak that usually harvested at 60 or 80 years old. Is I think it's quite interesting to be studied. But usually we found like defects with fast-growing tropical timbers, and we try to see that if this particular clone has the same problem or not. And the second question is, why is it called Jati Biotrop? Actually, I think people usually call that name because of the producer of the teak clone is the Seamio Biotrop. You may search that Seamio Biotrop, and because some names are mentioned, no, I heard some other names for this clone, but because I see the other researchers using the name Jati Biotrop, so I continue to use Jati Biotrop for marking that this was the same clone that is used by the previous researchers. And did I compare with the other tick, yes, I think maybe I rushed a little bit in the presentation, but actually the properties is quite comparable, and some properties are even better than the conventional tick. I think that answered the question. Is it? Or is
Okay.
or you have the follow-up?
Ibu Yulianti, do you have a follow-up question? Okay. Thank you, Mr.
Yes,
Raffi.
thank you, Ms. Rafi. It's quite enough.
Thank you. Okay, anyone, do you have any question? You can raise your hand. Okay, okay, there is a question from Professor Grill to Mr. Rafif. Why do you name the strain internal residual?
Okay, so Professor, this is like a test for me, but I believe that I have read some publication that to different to make a difference between the surface. So so I use the internal as the uh, name, and why still using the residual strain, because I believe that's not the maturation stress that on the surface, so it's kept on the timber.
Okay. Mr. Grill, do you have any response?
Yes, no. So when you measure the relationship between internal residual strain and properties, is that properties of wood near the surface or not? Or also inside the the wood, inside the tree?
So I took the specimens of basically near of every measurement points. So
Okay. Okay.
so for the internal one, I took near the internal strain gauge. For the surface, I took on that.
Yes, but then if it is internal measurements, you have to, well, the internal residual strain that you measure with the strain gauge does not correspond to the gross strain during the maturation. It has been, because it has been modified by later loading of the tree. So it does not really correspond to the maturation strain of that point. So it's okay when you do that with the surface strain. Okay, if when it is near the surface, it works. The correlation can work. But if you consider the strain measurement inside the plank, inside the tree, then it is not the real maturation strain that occur during the maturation process during wood formation.
Yes, I took a different correlation analysis. First is for the outer strains with the outer section of wood, outer section of the jati biotrop, and the second one is the internal section with the internal measurement of the strains. So, but because of the surface has no correlation, no significant correlation, I didn't show it in the presentation.
Okay. Thank you.
Okay, thank you, Mr. Rafif, and also Professor Grill. Okay, we have a question here from Professor Yamamoto. So he was a previous speaker in this in their series 1. The question: how about how do you think whether the maturation of wood quality depends on tree diameter or age? You can know it from the radial distribution pattern of some quality parameters.
Okay, I'm trying to answer Yamamoto Sensei.
Okay.
I think I was comparing this result with the model from an Indian scientist, but at all, I don't know if it's she or he, but I think the model shows like factor of age than the tree diameter. So I believe maybe in trees like thick, it's more of the age that affecting the maturation rather than the diameter. That's as short as I know for the moment.
Okay, Professor Yamamoto, is it enough for you?
Yes, thank you very much. We can continue to discuss about it later. Thank you.
In your room.
Yeah, in my room. In our room.
Yeah.
Actually, we are facing each other right now.
Thank you.
Okay, thank you. Okay, there are questions from Professor Imam Wahyudi to Professor Grill. Is genetic factor has specific effect on biomechanics of land? And also to Mr. Rafif, should be well considered in order to evaluate the timber quality, residual stress or longitudinal stress. Okay, please, Professor Grill.
Yes, thank you for the question. So the genetics has an influence on wood properties as well as environmental conditions. So the wood properties are result from both. Considering the biomechanics aspect, it depends what we talk about. Of course, wood properties are important for biomechanics, but if you consider the maturation strain, which is the source of growth stress, it might have a genetic origin, but the main is really the environment. It is one of the properties that is most linked to the environmental condition, to the growth condition of the tree. So like reaction wood formation and things like that really depend on the growth conditions more than the genetics.
Okay, thank you.
Thank you, Professor.
Okay, the next question to Mr. Rafif.
Okay, this I feel like this is a test for me from the other professors. Which one should be well considered to evaluate the timber quality, residual stress or longitudinal stress? Actually, longitudinal stress is just the direction marker of where the stress is oriented. Residual stress is, I mean, when there is a wood that still looks like wood, it still keep the residual stress inside, I believe. So both the surface or residual is important. Surface, I think, is we try to observe before the felling. We try to predict whether that tree has the high stress or not. So eventually, when it fails, we can predict whether there will be a defect or not. But the residual stress maybe is more important because the usage of wood will be longer and the residual stress is basically kept inside, and we have to know that as well to prevent, if we can, the defects that maybe occur because of the residual stress kept in the timber.
Okay, thank you, Mr. Rafid. Gambatte kudasai.
Gambarimasu.
Thank you, Professor Imam, for the question, and also for Professor Grill and Mr. Rafi for the answers. Okay, we have another question to Mr. Rafi from Mr. Romi. Jati Biotrop is actually cloned several times. As far as you know, will it affect wood quality for each clone? Will there be a different wood quality between the first clone to the next clone?
Okay, so the term clone, it should be has the meaning have the same quality because of clones, but the possible difference between the first clone and the next clone is maybe because of the environmental factors. Because of course the first clone and the next batch will have the different environmental or different situation, but the genetic should be the same if we talk about the genetic factors only. I think that basically the idea of why are we using a clone, because the quality should be the same for everyone, for every clone.
Okay, Mr. Romy, do you have any follow-up question?
It's okay. Yeah, it's okay. It's fine. Actually, if we talk about the questions, actually, there is something called in the we call it in for our clones things in our biotechnology, we call it genetic instability. If we clone several times, if we use something called somatic embryogenesis, or we use something called callus, callus development and everything, we have something called in genetic ability. We know the clone that will how to say, like, the genetics will be the same for each clone, but if we do that several times, sometimes just something some genetics something changed in their genetic in their genetics, because genetics will be changed if they always yeah, that's the main point actually what I would like to discuss, because sometimes we have to repeat the clone, we have to restart from the first again, and then we restart from the new ones. But that's only the things from the biotech side. But that's why I would like to ask: is there any proof that whatever you do in cloning, whatever you do in the propagation, it will never affect the wood quality? The wood quality will be the same. Will be similar. Yeah, will be similar with the first. With the first, because this clone, the map is like this. So whatever you do, the wood quality will be the same with the map. That's why what I would like to ask actually. Thank you.
Okay. Do you have any response, Mr. Rafi?
Oh, so for that question, I don't know how to answer, because yeah, we don't observe every clones, so I don't know. Maybe it's an interesting a research question for future research.
Okay, thank you. Anyone, please raise your hand if you have any question. You can also directly in this Zoom. Actually, I have a question to Professor Grill. Professor Grill, what would happen if a tree did not experience gravity relating to the growth stress or anything? Yeah.
I don't know. I have not. Thank you for that. That's a very interesting question. Thank you very much. Actually, well, I have not studied what happens in the space when they try experiments in the space. Maybe this could be observed, but in principle, the tropism of trees is light, is gravity, is the opposite of gravity, the light, and the tendency to be straight. So if you don't have the gravitropism, you will still have the phototropism toward light, and the tropism to a tendency of a stem to be straight. So it will be a very different. You will end up with very different tree shapes, only motivated by the light and not by the gravity. So it will be very strange, but it is possible.
Yeah.
But, yes.
Thank you, Professor. Okay. Anyone? If you have any question, please raise your hand or type it on the chat box. Maybe we still have like 4 minutes. If you still have a question.
Yes, I can answer the coming question about growth stress in bamboo. Yes, they do. I don't know exactly what tissue they have, but they do have growth stress. It has been measured. I have seen some Japanese colleagues who measured the growth stress in bamboos. As soon as there is an evolution of properties, you may have, well, bamboo doesn't work with wood layers like trees. But you have internal thickening of the cell walls. So there is no reason why there would not be residual growth stress. Maybe not in the same way as it occurs in trees. And it does happen, and there is growth stress. It has been measured, yes. It was work by Nomura many years ago in Japan.
Okay. Thank you, Chang Hua Fang, for the question, and also Professor Gill for the answer.
Hello, Joseph.
Okay, please.
Joseph is someone. Nice to see you here.
Hello. Very nice. Yes, thank you.
Do you have they have the published articles on the growth stress of bamboo?
I don't know. I remember when I was a PhD student in Uji, near Kyoto, there was a professor named Nomura who did this kind of test. And this was high. So I don't remember the publications themselves. But if you look through Nomura, he might have published in Japanese.
Okay. Recently, I did some research on bamboo growth stress. I find some very interesting things. Maybe next year in the 11th Plant Biomechanics, we are good there to present my findings.
Oh, very good. I look forward to it. Thank you.
Thank you.
And to see you again.
Thank you, Mr. Chang Hua Fang. Okay. Is there any question from other participants? So you can speak directly in this forum. Okay, I think there is no other questions. Okay, before we end this Q&A session, this is from Matsuo Sensei. There is a link, maybe related to the publication. Yeah, could you explain? Yeah.
Yes, this is a publication by Dr. Nomura, who was mentioned by Dr. Joseph Grill. It was, yes, as he said, it was in Japanese. So, but now I think you can have a look and you can translate by using AI translation for some content.
Thank you very much. I will check it. Thank you.
Yes, it's nice.
You're welcome.
Yes, thank you very much.
Thank you, Professor Matsuo, for the additional information. Yeah. Okay, before we end this Q&A session, if you closing statement, maybe Professor Grill and Mr. Rafif, please just short statement.
Thank you very much.
Yeah, maybe. Yeah, if you have any statement, please.
Okay, thank you very much for the kind attention, and I'm still learning, and I'm hoping I can do my best in this study. Thank you.
I would like to congratulate the organizers for the very nice organization of this system, of this webinar. It's very well done.
Yeah, thank you, Professor Grill, and also Mr. Rafif. Okay, concludes our scientific session for today. And on behalf of the organizing committee, I would like to thank all our speakers, including Professor Grill, Dr. Yu Cheng Long, and also Mr. Afif, for their time, knowledge, and inspiring talk. And to all participants with us today, I hope you enjoyed the session and gain new insight into plant biomechanics, from the cell level to the structure of wood itself. Okay, thank you. And I would like to return the session back to our MC, Dr. Siti Hanifa. Thank you, and thank you very much for everyone, and next time. Yeah, thank you.
Yeah. Thank you very much for Dr. Hairi Cipta for guiding this session, and also thank you for all the speakers for their insightful lectures. And also, I would like to thank the participants for the very engaged discussions. And please give appreciations to our distinguished speakers, moderators, and also all of the participants contributes in this session by sending reactions to the Zoom room. Thank you very much for all of us. And as a token of appreciation, we present the e-certificate to the speakers and moderators. First is for Dr. Joseph Grill. Thank you very much, Professor. And the second is for Dr. Yu Chen Long. Maybe he already left earlier. And also the last is for Mr. Vipul Jasmar, Master of Agriculture. Thank you very much for your contribution in this session. And before we close this session, please don't forget to fill out the attendance form, as we will deliver the e-certificate as participations through email. So our organizing committee already distribute the attendance form in the chat box. And we also would like to remind you our upcoming event, the 11th International Plant Biomechanics Conference 2026, which will be held in Yogyakarta on August 23, 26, 2026. And we await your participations in this session. And last but not least, for the participant, we also would like to present the e-certificate for our esteemed moderator for guiding this session, for Dr. Hairi Cipta, very much. And for the participants who haven't joined us for the previous webinar session, we provide the recording, which you can access in our official channels. You can scan the QR code and you can see the previous webinar recording session. And let's commemorate our session for today with photo session. I would like to invite for all the participants to open their camera. Yeah, thank you very much. And I will count on 3 for the organizing committee to screenshot. Okay, are you ready? Okay, I will count 1, 2, 3. Okay. Okay, please wait. 1, 2, 3. Okay. Okay, thank you very much. And that's conclude our session for today. And as the master of ceremony for today's session, I apologize for any inconvenience in guiding this session. And thank you for your participation. And see you again this year in the 11th International Plant Biomechanics Conference 2026. Thank you very much, everyone.
Thank you.
Thank you very much. Thank you.
Thank you.
See you next year.
Terima kasih. Thank you.
Thank you.